All right, now we are filled in. So our essential question: how does the sequence of DNA determine the structure of proteins and the traits of an organism? So in our past weeks, we did the structure of DNA, we did DNA replication, and then last week we did transcription and translation, where DNA got transcribed into RNA and then got translated into a protein. But what does that actually mean for us? How does that translate into we have brown eyes? That is what we are going to talk about for the end of our chapter. So the way proteins differ from each other is dependent on the number and sequence of amino acids that are translated from our DNA. Which was the process that we talked about last week. All right, the genetic code. You guys don't have this picture, but there's plenty of pictures out there like this. We'll get to it. So how do we know which codons match with which amino acid? We have to be able to read the code. The genetic code is a chart. That chart right there on the top right of all 20 amino acids found in living things. This is not something you guys will have to memorize, but we are going to learn to read it today. It is the number and order of these amino acids that determine which proteins are expressed. And the proteins that get expressed translate to the genes that we see about ourselves. So we compare this to the alphabet, the English alphabet anyways. There are many different alphabets all over the world. But in the English alphabet, we have 26 letters. But out of those 26 letters, we can make millions and millions and millions of different words with just those 26 letters. And we do that by putting those 26 letters in different orders and different lengths. The letter I can be a letter or it can be a word all by itself. That is a one-letter word. Or you have the word anti-disestablishmentarianism, which is a lot of letters. So we take those letters and we mix them up in different ways and we make different words. That is how these 20 amino acids work. We take these 20 amino acids, they get translated in a different order for every single person because all of our DNA is different. And different proteins are expressed. So we're going to take an example codon. Remember, codons are the groups of 3 on the mRNA strand. We always read them by the mRNA strand. And our example codon, we're going to do AUC. So we've already gone through the DNA. We've made our mRNA strand. Now on that mRNA strand, we're going to take 3 sample letters. And this is how we read the genetic code. The first letter of our codon is A. So the first letter is found on the left side of the table. So here's our left side of the table. We go down to A. And now we have a row that we're looking at. We don't have to look at any of the rest of the chart. The second letter in our codon that we are using is U. And the second letter is found on top of the table. So here's where our U is. So now this is the column that we look at. So now we have a row and we have a column, and we look at the box where the 2 intersect. So here's the row for A, the column for U, and here's the box where they intersect. Now you can ignore the rest of the chart. You're only looking in that box. And then the third letter in our example is C. So now we go to the right side of the chart. Now on the right side of the chart can be a little tricky because they have all 4 letters. But we have to go all the way down to our row that we've already established, into our box that we've already established. Our third codon is C, so there's our C in our correct row, which points to, there's the AUC. And all charts are different. Not all charts have the codons like this. This is just an example one. On your quizzes and tests, the codons won't be in the boxes. So now, if we just take that one box and blow it up, you can see AUC equals ILE right here. That is the actual name of the amino acid that the codon AUC translates to. So we read the square where all 3 letters intersect, and AUC equals ILE, which is just abbreviation for isoleucine. Again, not something you're going to have to memorize, is the names of these 20 amino acids. You just have to know how to read the chart. All right, and then you have an amino acid chain that equals a protein, sometimes called a polypeptide because they're bonded together with peptide bonds, but more commonly called a protein. Like I said, there are only 20 amino acids that determine every single one of our genes. By putting them in different orders and different lengths, just like words and sentences. Our genes are determined by the number and sequence of amino acids that are translated by these 3 processes: replication, transcription, and translation. So that is the end of translation. And we put this together with gene expression. So how do those proteins actually express our genes? So we move on to 8.6. Gene expression is carefully regulated in both prokaryotic and eukaryotic cells. If you don't remember what those are, prokaryotic cells are just very, very simple bacteria. They don't have the full nucleus. They just have the DNA floating around in the middle region called the nucleoid region. And then eukaryotic cells have the full nucleus separated off. Their DNA is separated off into a different organelle than the rest of the cell. But once these proteins are translated, these big long chains of amino acids, now what? So gene expression, the order of proteins at the end of this process determines which genes are expressed. So the gene for blue eyes has a different order of proteins than the gene for brown eyes. So we just went over all these processes. DNA, transcribed into RNA, translated into proteins. At the end, you have a big long chain of amino acids that makes a protein. Everybody has a different original DNA sequence, so their genes that are expressed in the end are going to be different. Cell differentiation. So all of our cells have a complete set of DNA. We have 3 billion base pairs in every single one of our cells. So why aren't all of our cells the exact same? It's because only a few genes are expressed in each one. Out of those 3 billion base pairs, this part will get expressed in your eye cells. This part will get expressed in your stomach cells. This part will get expressed in your skin cells. They're all there. All 3 billion base pairs are there. But only certain genes, remember a gene is just a piece of DNA, basically get turned on so that our different cells express different traits. This is called specialization. So differentiation is the process of making all those cells different, and then specialization is the process of making each of those cells special. Kind of the same concept. Prokaryotic cells is what we study because it's very easy to see and to study their switches. Just like a light, turns on and off. They're literally called gene switches. So prokaryotic cells turn their genes on and off like a light switch by controlling transcription. And like I said, we have 3 billion base pairs, and a gene is just a piece of DNA. A promoter is a DNA segment, a piece of that DNA that allows the gene to be transcribed. That's basically the on switch. And then an operator is the part of DNA. That's the actual switch that turns the gene on or off. Promoter is the start, like the start button. So in the picture, you can see just that piece of DNA at the beginning is the promoter. And transcription will happen just like we learned until they hit a codon that says stop, literally a stop codon. Until termination happens. So out of those 3 billion base pairs, only a piece of that DNA is being transcribed, not all of them. And it depends on what the cell needs. Our bodies and prokaryotic cells, they don't waste time transcribing genes that we don't need. It's a waste of our energy. Eukaryotic cells, we are actually more complicated. They have more than one switch to control the expression of genes. So just like in my room, I have 4 light switches. I can turn on the front lights, the middle lights, the right lights, the left lights. And depending on how much light I want in my room, I only turn those switches. That's what our DNA is like. Different sets of genes are expressed in different types of cells because sometimes they're flipped on and other ones are flipped off. And then transcription is controlled by those specific DNA sequences. There's a lot more detail to this that I actually go over in my college class, but for now, genes have switches. They can be turned on and off. Not all 3 billion base pairs get expressed at the same time. We only express those genes that we actually need, or else it's a waste of our energy.